It's on. Okay, so yes, really today we are already doing it. So we assume that we already, let's say, tackled this part of the short syllabus. Recent advance in genome technology. I don't think it's really needed to go into the technological detail, but if something is not clear, feel free to ask. Here, now, later, you know how to reach me and grab me. So a lot of data. Basically, the take-home message is now we have the power. Everyone has the power to do genomics with a bit of resources, and but then there is also a big deal in data analysis. So if you can get the joke, it means that you've been doing quite a bit of scripting. If not, lucky you. Okay, so now we go really into example, an applied example. So what I will be presenting you, this paper, which is both a paper, but is a really nice example of translating science for end user. It's a research effort from scientists at CSIRO, so the kind of NOAA, but of Australia and Tasmania. They are really, really advanced in marine research. So if you still have to plan your research period abroad, think about CSIRO. And they produced a leaflet called Oceans 11. Yeah, that's what I'm saying. At that time, George was still, and now it is a movie, let's say, how do you say, a series of movies? Because then they did Ocean 12, and then Gender Balance, and so on. But you all know that even the one with George Clooney and Brad Pitt is a remake. The very first Ocean 11 movie is a movie from the 70s with Paul Newman. So the very first one. So they call it Ocean 11 because you need also a catchy title, because they decided to summarize 11 applications. They could be 10, 13. They decided to stick to 11 also to say to make it really to make it really impactful. So it's a review paper. This is the abstract. These are the authors. You can say mostly from Australia. Jennifer Ovenden is a fish population geneticist, not only in fishery science, but also in conservation. She's the kind of queen of research in Australia, as Barbara Block is in the US. I don't know if you ever heard of Barbara Block. Okay, don't worry. It's a quite tough review paper, almost 30 pages. Imagine you go to a fishery manager, you say, yes, I have the solution for you, isn't here? 30 pages of academic jargon. So they translate it. That's the movie you all maybe remember. But that's the real one. That's the original. The first Ocean 11, no? And no, it's not Paul Newman, sorry. It's the singer. What's the name? Now, questo è quello che faceva anche il cantante. Vabbè, adesso mi verrà in mente. Frank Sinatra. And they translated into a field guide. This is what I want to transfer to you today. We only have one morning, so I don't want to enter into the very cutting edge approaches, but I really wanted to convince you or raise more awareness, if you are already convinced, that genetics can be useful in marine research. Sorry, I have a strong focus on animal. I belong to a zoologist group, even if I'm not a zoologist, so fishery. So fishery and aquaculture. I want to present you example applied to algae or plants. So I admit my bias. And I also have a preference for vertebrate rather than invertebrates. So most of the application are on fish. So you see, they translate the into 11 application under 4 category, which is understanding what we harvest, understanding the impact of the harvest. Harvest is fishery or aquaculture can be seen as a harvest too, and then biosecurity, and also post-harvest regulation. If any of the word I'm using it, you don't understand the meaning, or you don't understand which meaning I'm using, because harvest can have different meaning in different research field, like recruitment, no? In ecology is something, in fishery science is something else. So please stop and interact. But please stop and interact for any reason. So species identification. It was also the first of your input. Eyes over there. Said we have to identify fish. Now, come on. We are all super good in identifying our target species. Are you also sure you are so good in identifying when you find their larvae, or when you find their remain in a gut content, or when you have to deal with the juvenile that still didn't develop the diagnostic feature that you are searching the dichotomous key? No, so it's not that easy to identify morphologically, or let's say phenotypically, because it's not only external morphology, can be vertebral count that you cannot get from the external. It can be also some physiological parameter like spawning period or other behavior. Nowadays is what is called DNA barcoding. Barcode is any feature that you can standardize to identify. So we have barcode in the products, we have barcode for our flight ticket, no? Anything that can be standardized, easy to read and transfer without modification, and contains information. And this is DNA sequence. So DNA content is the same if we forgot about epigenetics. If we are really thinking about the sequence of the nucleotide, so the genetic code, the one that is called the instructor, now the book of instruction to build the species, is the same, even if you take a larvae, not the egg is only half, but you have to take the post-zygotic life stage, or the whole individual from every somatic tissue. Okay, so you can identify, and this methodology has a limitation. I work with mostly molecular tool, and other tool. No tool is the bacchetta di San Buco. Is the magic, most powerful one on Earth. Okay, one tool is never enough. Every tool has limitation, so we have to integrate. And again, molecular taxonomy has limitation, Now we talk about integrative taxonomy. So we couple molecular with phenotypic and also ecological data. And this is, you see, I kept Genodream experience everywhere, even if now we are not anymore Genodream, we are Beam Lab. So again, fishes don't come with the ED. Also humans, sometimes they don't have ED or they have fake ED. So, but it's not that easy to tell the species. It can sound trivial. You can think of me like I'm a very hopeless. How come on, you cannot identify your species? Can you identify species from fillet like that? And you have to detect fraud. Or yes, you can tell it with molecular data. So in most of the cases, not in 100%, sequencing some specific region of the genome can provide you with robust information about the species this specimen can be associated with. So again, DNA barcoding is not discovering a new species. You need to have the standard. You need to have a reference sequence. If you have to discover the composition of a compound, of a metabolic profile, you need to have the standard. You need to know all the different reference, and you can only assign your unknown sample. And this is how molecular taxonomy works. You need to have the reference library for all species on Earth. And this is what the International Barcoding of Life Initiative is doing since 2004. It's a worldwide initiative, started from Canada. It's not based on US, Paul Ebert. Started because they realized that traditional taxonomies were in danger, are in danger, because they are retiring, they are passing away, and no one is updating their knowledge, because it's the knowledge of a life. Do you remember the images of drawer full of butterfly, of insect? Who can recognize? Now we have AI, computer vision, but still you need something which you can rely upon, which is identified in the proper way, is already there, you can always come back and check. And if you produce a molecular standard, a molecular reference from what is called voucher specimen, then any other unknown individual that is 100% equal to that, then you can associate the same species. And this is the same that we do. Now there is a sub-initiative of the International Barcoding of Life, which is MARBOL, Marine or Fishbowl. Okay, so this is the web page. DNA barcoding works quite well with eumetazoa, so animal, the way you think of an animal. There are some limitations for, especially for elasmobranchs. They have a very slow mutation rate. They are a bit peculiar, so sometimes the standard marker for barcoding of life, which is a portion of the cytochrome oxidase 1 mitochondrial gene, doesn't work. So you have to use a multi-marker approach. You have to integrate with another marker. This is quite well established in the scientific community. So this was the very first European project on this topic called Fish and Chip, because at the time the fashion genetic tool were microarray or called chip. So again, Fish and Chip. And from what we learn with this project, we are now carrying, since 2008, let's say, autonomous, spontaneous project. It means no dedicated funding, we managed to run it with the savings from other funding, with a sort of crowdfunding from the Research Institute before it was fashion to do crowdfunding, to create the reference library of what? Of all elasmobranchs of the Mediterranean. Why elasmobranchs? They are treated, but they are also target of some fishery. And even though there are less than 100 species of elasmobranch in the Mediterranean, taxonomists like to change, like the stairs in Hogwarts, you know? They like to change. They change names, they change systematics, and so it's kind of messy, also because elasmobranch are associated with a kind of evolutionary stasis. They call it live fossil, no? Like shark are in the shape of sharks since a long time. So it's not easy to identify correctly elasmobranch, especially juvenile. And which are the elasmobranch that are mostly caught by fishery activity? Juvenile. So that's why. So this platform is the barcoding of life platform is very well conceived, is very well curated. Canada, but also other foundation provide resources to have it maintained. So it's free. You can use it for free, but there is real human being behind that are checking. And we get emails sometimes from different people say, you entered the record, you assigned it to Spanish water, but it's a kind of the border. Are you sure about your coordinate? Are you sure about your picture? So it's really curated and you can trust. And what we discovered with this first attempt, we were working with I did participate to surveys which are not existing anymore because I'm old. I did the Grund survey. Who knows what is Grund? Because you work with old people like me, no? So it's one of the very, it was the winter survey of fishery monitoring. Now we have MERITS, Mediterranean Roll Survey. If you do FishMed, you should know what is DRCM, what is ICES, what is MERITS. Let's say there is and so they are troll survey because still most of the fishing effort is by trolling. And we were working with all the groups that were carrying out the survey, and they were experienced people doing those experienced research and doing very seriously, very dedicating a lot of effort, but still a lot of misidentification were occurring, even by a really knowledgeable research group. So, for example, you see that all the torpedo from the Levant, these are found division to make your life easier. So it's Western, Eastern, and the main FAO division, they were misidentified. And when you provide the landing data, any data, and you associate it to the bronze species, there is no way that you can then reconstruct the picture. So you see, it was at the beginning very complicated. So it was a very collaborative effort. We went back to the colleague and they say, thank you. So let's try to understand why. And then in most of the cases, we realized that the protocol for identifying species were based on adult individual. And for example, some of the dermic pattern or some of the feature were not evident in smaller size individual. And then we could apply this also to fish larvae. Who is doing with acoustic? You get larvae. Passive acoustic. But a lot of fishery model are based on larval data. So there are a lot of larval survey, and this survey provide the data. So based on the larvae you found, now I'm not good in model, but you are using model, no? Larval disperser is like Lagrange, but larval survey, they count the larvae and they predict or estimate the recruitment, the biomass. I hope I'm not using the wrong. But what happens if you take a larvae and you consider it of Tunus tinus, and while it's of Tunus alla lunga, no? Obviously, you say, who cares? No, it's a tuna, but Tunus tinus has quota, Tunus alla lunga is not yet. So that's again a different value. And again, what was the problem? That again, the protocols, it was not, the problem was not someone that was not doing his job properly. Everyone tried to do his job at the best. But the protocols for larval identification were based on the wrong reference material. And we can identify even egg case. Everyone has seen this. How do you call it? Mermaid purse, Borsellino delle Sirene. Have you seen this on? If you find a lot of this, what does it mean? There is a... Yes, but a lot. If you find the aggregate of egg case, there is a nursery or a place where they go and deposit and lay eggs. And again, it's very difficult to identify from morphology the different species. They all look like the same, no? But then with the biological remain, which are inside the egg case is, I don't remember the material, but you don't extract the DNA from the egg case, but you extract the biological material from the leftover. Obviously, is the biological material of the mother. So you can tell the species of the mother. Every time you work with mitochondrial DNA, you can only tell about the matrilineal. So if there is an hybrid, you cannot tell. And hybrid do occur. So again, this was very effective in to identify a potential nursery of Rhaiasterias in the Sicilia, I cannot say it, nel canale di Sicilia, per una bolognese difficilissimo. So the other point, okay, species identification, again, I tell you, is not easy, especially when you are not talking about fish, when you are talking about invertebrates or sponges, for example, is very complicated. And it's also very complicated also from a molecular point of view with the sponges of some invertebrates, because they are more challenging. They don't want to give you their DNA so easily as vertebrate. They have inhibitors, and also they lack a good reference database because they are kind of not easygoing species to work with. So there are fewer groups and fewer data, but there are ways to at least identify a specimen. Please don't say, Cariani told us that with DNA barcoding we can identify species. No, identify species, you need to really describe the species. You can identify specimen, 2 species. Okay, this is a few points that I cannot. And then we can, given that you have one species, different population of the same species, let's say, you can identify boundary. We love boundary. Now we want to put boundary everywhere on Earth and in the sea. But the marine animals don't care about our boundary. They feel other boundary in the sea. The wrong assumption was that in the sea there were no boundary because there are no mountain, no river, so the fish can go everywhere, reproduce with everyone. So the very famous statement of Thomas Huxley at the Royal Academy of London. So Thomas Huxley was a supporter of Charles Darwin. So it was not the first stupid talking, no? But even Thomas Huxley, he said that the fishery resources were endless because they were all permitted, no border in the sea. And so they constitute a kind of endless resources. Now we know that fish are not the same. There are boundaries, there are locally low connection population. And if we harvest too much one population, it cannot be replenished by the other. We can go toward local eradication of a species. At that time, the information they had was not, and also the fishing pressure they had, it was not the case. So this is where most of our research have been digging over the past year. So we, but also other research groups have been doing a lot of population genetics. I don't enter into population genetics because tomorrow you will have population genetics of humans. So what we do with non-human species, we copy what has been developed in a human, and we fine-tune it for our species. So you see a lot of projects, and where are those projects more important? When we face shared resources. So, for example, the Adriatic Sea. There are at least 4 countries fishing in the Adriatic Sea. Italy, Croatia, mostly, but we have Slovenia, few fishermen, maybe, Montenegro, Albania. So we have to define, and we cannot tell up to here is my fish, now is your fish, okay? We need a shared management, and we need to know where the biological boundary can be defined. So several projects have been funded by the European Commission, by the GFCM, by ICES to check where the stock boundary, stock is a management concept. The stock is the population which is harvested in a very easy definition. But does your stock, the stock that you define, truly align with the biological population? You can insist on a mixed stock. So you are fishing in, for example, a feeding ground where different biological population come at certain time of the year for feeding. But then they are separate for reproduction. So biologically they are not permitting. Permitting means that everyone reproduces with everyone. And that you have to take into account. Or you can consider your stock limit, but then maybe your biological population extend over those limits, and it's just one biological population that covers a wide area. And then it means that everyone has to sit at the same table and make an agreement. So that's why all these projects have been funded where there was a shared resource. In the Adriatic, FishMed is small pelagic between France and Spain. The Western Mediterranean Sea. Transborane is the worst name ever for a project. All the emails were blocked by Univo spam, and is the transboundary population structure in the Alborn Sea. Again, a sea where we have not only European countries, we have Spain, France, Italy also go fishing there. But we have Morocco, Algeria, and Tunisia. And what genetic can tell? Genetic can tell that fish are not the same. Fish can be locally adapted. Fish population, sorry, can be locally adapted. They can develop unique molecular fingerprinting that we can analyze, and then we can trace back. So we can trace back not only to the species, okay, this we can do it low cost quite easily, but with a bit more effort, we can create a reference not only for the species, but also for the different biological population that constitute the species. And if we develop a tool that it's just a matter of sensitivity. Having a genomic approach can give you more power to develop a tool which is more high resolution. It's just to count the otolith ring, and if you only have one magnifying lens, or if you have more, no? And you can fine-tune what you need for your purpose. So this means looking for specific region of the genome, what we call outlier. Outlier is just something that falls outside, no, your distribution. And with the outlier approach or other genome scan approach, you can really define more with a higher resolution the geographic differentiation of the population of your species. Solea is a nice example. They are not that great swimmer. They are lovely flatfish, but yeah, they can swim not long distance, but they have larval dispersal, so that they can have good connectivity. This is what you get with normal approach. So Western Mediterranean is all the same, Adriatic is all the same, so it's a large scale picture. If you develop through genomic more tailored, if you have with genomic, you have the potential to develop more tailored approach, and then you can really, for Solea, trace local population. So you see, Adriatic is not the same, and the border in the Adriatic is not horizontal as it is, but is vertical. You see, it's different from Eastern and Western Adriatic, and the same for the West Med. So you have a high resolution, so you can not only trace the species, but you can trace the origin. And why this is important? Because you know that with fishery adaptive management, some area can be close to fishery in some period or for always. And you have to declare the fish that you are selling where it was caught. And then you can check, do you really, if you declare that this fish is from Adriatic, if there is a doubt, if there is an enforcement, so show me. I can tell you if it's really from the Adriatic. Okay, you understand the potential application, no? And if you want to do stock assessment, does it really make sense for you to run stock assessment for GSA 17 separately from GSA 18? You know where the GSA are? Let's say North Adriatic and South Adriatic separately? Kind of. You should do North Adriatic and the eastern part of the South Adriatic. No? So you have to reframe your stock assessment. And once you did, why this is important? Because this is the molecular, say, the definition of the reproductive unit, because DNA gives you the evolutionary perspective. So DNA gives you the reproductive interaction, no? One generation after the other. Then you have parasites that are more short term, that can really tell you what you have on you, and that also they have distinct distribution. So it's just different layer of information. Each layer is relevant to really define the stock units. So you see, these are all, let's say, oceanographic, life history. So what you get from survey, from fishery survey, so growth parameter, biomass. You can put on top also fishing effort, and then you have genetic. And it's just one layer, and all the layer matter because they give you a different perspective. And then it's a matter of finding the proper way of integrating all this information. And if you find that there is a mixed stock, how you manage, again, for example, tuna or anchovy. Tuna, you have quota. You have quota for the eastern and western stock. You all know the situation of bluefin tuna, no? A huge depletion, high commercial value, high economic value, which as a cascade effect is linked with political and social aspect. No? Who is buying tuna in the map? Mostly the Eastern, Far East market, and so on. And so for tuna, we have, for tuna and other species, we have a dedicated institution, ICAT, International Commission for the Conservation of Atlantic Tuna. Doesn't only care about tuna, cares also about other tuna-like species, for scomber, for small tuna, for blue shark, so for mainly big, medium pelagic. Tuna is quota. And how can you define the level of quota assigned to the eastern and western stock? And what is the boundary? 45 meridian, in the middle of the Atlantic Ocean. Is a boundary drawn by manager, by politicians. Now, so we have quota for the Americas, so Canada, US, and every fleet that buy that quota. We have quota for Eastern, no? European, but also North Africa, Morocco has quota, and other, Japan also. They both quota, and they can fish. So, but fishery does not occur inside the Mediterranean or Gulf of Mexico, which are the supposed spawning area. Fishery occur in the middle of the Atlantic. So you have to determine to which extent each fleet insists to the two stocks as they are defined. Tuna are not solea. So tuna, they do move, they are bed spawner. Wherever they want, they find a nice place and nice situation, they spawn. So they have a very long spawning season, they can spawn multiple times, so it's very complicated. It's nearly panmitic. But still, so a lot of study before and they found no difference. But there are phenotypical difference, so different growing parameters, so there must be some different. We really needed genomics, so the power to really assess genome wind to find some portion of the genome that do have the signal of a slight, low but significant separation, differentiation of the two stocks. And once you develop the tool, once you find the correct magnifying lens to tell the difference, then you can assign. You can assign fishes to one or the other. I'm making this very easy, but I don't want to make it trivial and stupid. There is a lot of science behind, but I don't want to enter into technicality because I really wanted you grasp the potential, not the single approach, especially in in genomics where the approach are changing so fast. Is still the end? No. With tuna, it will never be the end. It will be more complicated because the species is challenging, but it's also kind of cheating on us because all the models, all the assumption, all the stock assessment assume two spawning population. And now we are finding larvae. Now that we can identify larvae of tuna with molecular, they are finding larvae in the Azores. Outside Gulf of Mexico, in the Slope Sea, they're finding larvae and spawning individuals, so adult with gonads. It means that really spawning is occurring here. So now that we have a tool that can define the two biological units, we discovered that these two biological units then are really intermingled. So tuna for sure will be always a species with more study to be developed. Tuna, and I mean bluefin tuna, but similar species with similar characteristic. Okay, and then what is also important when you do models? Classical ecological population dynamic. You need classes, no? You know the species, you can tell the geographic location, but it's not enough. You need to know the life stage, so juvenile, but the more accurate you are by year. And what do you, how this is done normally without molecular tools? It's done by length, weight, otolith, or other structure that can tell you the progressive growth, no? Everyone knows what otoliths are. Again, telling the age. Now you say, okay, come on, you cannot tell if this is an egg or another species. You are really honest. And again, you cannot tell me from an otolith how old is this fish. It's not easy. I'm not doing it, but people that are, who is reading otoliths here? Or bone rings, or I work with otolith, but not studying the age. I just work with the structural pattern harmony. Okay. But from I have colleagues that are reading otolith, and they say that it's not that clear cut, you know. So the concentric structure can be blurred. It's not that easy. And so there are a lot of effort between scientists to benchmark the methods. So that if different researchers in front of the same otolith tell us they provide the same output. And this is the benchmarking otolith or spine. Now the first spine of the fins of the dorsal fin is another structure, or bones is more time consuming, not to calculate age from bones. You have to take the bone. Afterward, we all go outside and take all the tuna bone that we can find, and again you can see it's like a tree. You have the ring. Again, from humans, we can develop tool methylation. Methylation, pattern of methylation can be correlated with statistical high confidence with biological age. And then it's a matter of chronological age, biological age. There is a lot of research in human, and I'm showing you on purpose this research paper because it's from a colleague at the University of Bologna. And Donata Yusufizelli will tell you more tomorrow. We discussed it yesterday. What are you presenting so that I cannot double duplicate the effort? And they got the patent out of it.
I know that Claudia Franceschi is the one by the inflammation, right? The inflammation theory. But when you age, your immune system
Right.
kills you, let's say.
Yes, he's a professor at the. I don't work with him. I work with Elisabetta Cilli for the ancient DNA and with Donata Luiselli. But since we are good colleagues, they were telling me this their work, so I decided to show their work on. It's called epigenetic clock. So can we transfer the epigenetic clock from human to other marine animal? Yes. Which animal will be easier for? Mammals. Human are mammals. So it was easier. And so we have a epigenetic clock for different species of killer whales or different species of cetaceans. For marine animals, which are not mammals, so it is more difficult, but there is a lot of research going on. This is really a tool which is promising, and we already have the first outcome for commercial species. So sea bass, obviously they are limitation because these are studies conducted mainly on animals marked recapture and or captive animal. So you don't know how because methylation is the interaction with the environment, and you need more data to be sure that the signal is stable, because methylation can change according to the environment and the life history of the animal. But for sure, epigenetic methylation is a really promising field to get from biomolecule, from the DNA, information that a few years ago, these papers are very recent, a few years ago were only possible with direct sampling of animal. If you want to read the biotomy of an animal, what happens to the animal? You can only do it once. With less invasive sampling, you can repeat it in life. Right? Think also of this. Ecosystem monitoring. This is another exploding field, I would say. For a decade, we have been doing stock boundary and species boundary. So identify specific species and do geographical region or biological population. You can have different biological population in sympatry. So in the same area, for example, you have the early spawner or late spawner of herring in the same fjord. They are locally adapt different conditions. They are to some degree structured, separated in their production, but they do stay in the same fjord. And now, let's say from 2014, so we are approaching nowadays, what we can do community level assessment. All the other applications, you need to have the tissue from one individual. Now you can do it with the mixed metrics. We call it complex sample. Again, we are doing in natural science what we learn from where? From human microbiome. We know the importance now. We are all of you. Everyone is an all of you. Every human living every living being is almost an all of you. So we have a different community living inside outside us, no? And so what was applied and developed for microbiome characterization is now applied to natural community with difference, with technological difference. So we can try to understand the relative composition of a complex sample. The complex sample can be a zooplankton net, and you cannot identify all the larvae or all the different life stages. And so you mix it, you put it in the milling beamer, you extract you extract the DNA of everything. You don't have to separate different organisms. And then there are methods to say, okay, in this milkshake of zooplankton, we have copepods, we have chetonates, we have all the different animal, or not only animal, also plants group. Sometimes we can identify up to species level. We have Physalis in this sample because maybe they were left over, or we can only stop at higher taxonomic resolution. I think it's the same also for microbiomes. Now, sometimes you can only give groups. You cannot reach strain specific. And in fishery science, this is really useful, for example, for trophic study. Everyone has ever tried to identify gut content? No?
Yes. When I go fishing.
So for
No,
you enjoy.
Yes, when I clean them.
But if you want to do trophic model, you need to know the prey-predator relationship, and you can only tell from gut content. And analyzing gut content is really complicated because in the gut content you have partial remain. And what you can identify in the gut content is that the part of the organism that are more difficult to digest. Gut content of Capodolio. The sperm whale. What you find in the gut content of plastic, gut content very much, but you find a lot of
Things are very sweets.
Sì, exactly. Now you find a lot of, you know that the sperm whale do eat cephalopod, but you don't find the cephalopod. No, cephalopod have no bones. You only find the backbone. Now I don't remember the proper name in English. In this case, okay, it's kind of easy. But if you have to do other, you can identify the prey from their otoliths. But again, you need to have a good reference of otolith shape to identify this otolith. This shape is for sure a sardine, or is a scomber, or is something different. Again, molecular cannot solve totally, but can have. One big problem when analyzing that content is that you have to get rid of the DNA of the host. But then it's a problem if you have cannibalism. So it's complicated. I'm telling the good part, but also the pitfall. No? But there is a lot of potential. And again, you can recognize some name. The most important person in Fano, Marco Stoggioni. In this building, let's say. But so gut content is kind of microbiome, no? Like you can also associate in the diet analysis, you can integrate morphological, stable isotope, and molecular. It also depends also on the rate of degradation of the biological remain inside the gap. And you can apply the same procedure to the environment. You can collect biological leftover from the environment. It's called environmental DNA. And then this environmental DNA is collected. And then the approach is meta-barcoding. Sorry, I forgot to tell you before. Barcoding is when you have one individual at a time. If you do meta-barcoding, is that given that now you have the potential to process a lot at the same time, you can do meta-barcoding and you sequence whatever, and then you try to assign to the different, let's say, owner of that biological domain. You can do it in the gut, you can do it in the composition of parasite, or you can do it from the environment. When you don't see the community, but you collect the biological leftover from the methods. You can do it from soil, airborne. Do you remember Expo a Milano, in Milan? There was a massive investment in environmental metabarcoding of water, soil, and air for the pollen, because a lot of plants were taken from the different participating countries, and there was a strong effort to monitor to avoid the spread of non-indigenous or any kind of problem. Obviously, if we do environmental DNA of this room, there will be maybe more biological remain of myself, because I've been moving a lot, I'm touching a lot my hair, I'm talking, so I'm speaking. Then no one is bored here, or nessuno ha pensato a capelli, speriamo Trevor. If Trevor Willis was here, less biological remain, because he has less hair and less way to leave traces. So that's the same for environmental DNA. Again, that's limitation. Not all animals release biological material at the same rate, in the same amount. So it's not perfect that. You cannot really tell the difference in quantity. You can just tell some relative abundance, but still it can help. Sentite anche voi una voce su? Sì. Ma gli diamo sicuramente più fastidio noi. And again, what elasmobranch, they are more difficult for environmental DNA because they don't release gametes in the water. They do have gametes, but they don't release it in the water because they have internal production. So for bony fish, sometimes you find a high quantity. For example, we have some samples in which we have a lot of tuna, and I say, where did you get them? They were slaughtering a tuna nearby where you were collecting. No, it was larvae or gametes. It was indeed a sample from the Balearic Sea, which is known to be an area of spawning of bluefin tuna. So, but again, environmental DNA has a lot of application, and you see also the skeptical optimist has a lot of potential in its application, but we must be aware of the limitation. And then you can also decide what you want to search. You can go and search for a specific species and you only want to know there is or there isn't, or you want to reconstruct the community and you just want present absence data, and then you can apply analytical method, or you want to have semi-quantitative data. So it's just a matter of your experimental design. And you can collect water actively. So go taking the water and filter actively the water, or you can do something else to the job for you. You can drop something at sea and you let absorb a sea. So it's less efficient. You cannot control the volume of the water that you are filtering. So quantitatively, but you don't need to be there. You can do the device to someone that goes at sea daily, who the sea operator. So fisherman, I would say sea operator in a more broad sense. Scientists, how often do we go at sea? A month, once a week. So let's give them tools. So you participate actively to a lot of survey.
Okay. Like to at sea underwater, like by land. We go at sea by land.
Okay. But so not all of you, not all scientists has a lot of opportunity to really go and sample at sea. So the more easy we find a way to collect information, it's the same. I think you already had this lecture, no? Michela Martinelli showed you the FOSS. Now what the FOSS is? Say the FOSS is FOSS means fisheries observation system. Okay. So they use fishery vessel as oceanographic campaign, no? And probably Michela, Dr. Michela Martinelli also explained the use of some, I don't know if they are collecting also DNA in some cases, or they do, for sure they do analyze that content with molecular. And so you can develop other devices and you can give them to really happy sea operators to use them at sea. You have to convince them, you don't have to impact their fishery, their activity, no? Otherwise they will throw it. And they do work. Linda can tell you, this was her thesis, master thesis, not PhD thesis. And they could work better, but they do work. They are a really powerful tool to reconstruct a community. Then you don't have to oversell or overinterpret, over-speculate the data. But this is a good practice in all field of application. But you can get a really meaningful result. And here is the work of another colleague. You see, I'm not doing anything. I'm just doing a report. Reporting the work of someone else. This is the effort. You remember that I started in the 2008 with the reference database of all elasmobranch of the Mediterranean. From there, we could move to environmental DNA because we had the standards, so the reference database of almost all species of elasmobranch in the Mediterranean. The one that we don't have is because they don't exist. Taxonomy is invented. Okay. We are fighting with taxonomies that we want that species to be removed from the checklist. Echinorinus brucus. No one has seen this species. Do you have sample? No. Or Glaucosterius salawi is that seems that is in the Mediterranean, or Raya africana, other species that really they are dream of taxonomies that wake up one morning, prepare a paper, and then blame on the editor that accepted the paper. And so this is the result. And we benchmark a lot of methods because we had no money to go at sea. We had to beg, pray, promise food, wine, and everything to our colleague going at sea to sample for us. And so we had to test different sampling methods that could be used on board by different research team. And we went to the Genova Aquarium to test all of them in parallel, to benchmark all the methods. Okay, there are some differences, but we can integrate them. And you see, we were able to, through a very large collaborative effort, to collect more than 1,000 samples. all along the Italian Sea. And what's our aim? To really help the definition and the establishment of ZRIA. Israel, sorry. Important shark and race area. And we can really see a pattern that the color are the UCN categories, so critically endangered, and so on, of the species that we detect in the different areas. So it's really something that has a limitation, but has a lot of potential. Okay, and now we move. These are the descriptive, not the attribute of where we are fishing. So which species, the boundary, the life history traits, so the age, the community composition, and then what happens when we need how genetic can help on the impact of fishing. Genetic can help really estimate the abundance, the spawning stock biomass, the number of individuals. And this is an approach that has been developed by Xyro. It's called close-skin marker recapture. Again, it's really complicated, the algorithm, but it's a very simple assumption. Marker recapture, we all know, no? You go, you tag, you go again, and depending on the number of the tagged individual that you recapture, you can estimate the census, the number of individuals. Clear for everyone? Okay. So this is marker recapture, nothing new. Close skin is like assess the degree of relationship, of kinship. So the more they are related genetically, the individual that you collect. So I always make this comparison. If you go to Bologna main square and you meet 100 people, people or person, 100 human beings, how likely is that they are related to each other?
Maybe they are not brothers, but they don't.
Okay, this depends on the mother, but if you go to Plan de Gralba nearby Selva di Val Gardena, and the surname of everyone is either Senone, either and any other surname is where my family goes skiing. I just go there eating, I go to ski, but my family. So how likely is that they are relative? So it is a matter of how the census of the reproductive community. Okay? That's exactly close-skin marker recapture. If you collect thousands of fish, the more related individual, like parent offspring, or half sibling, like sister and brother, you find, it means the lower the census. Okay? The higher probability to reconstruct this one, reconstruct sibship family, let's say the smaller is the reproductive population. So and the other way around, no? If you go to Città del Mexico and you get 100 human beings, it's very unlikely that they are related, no? Because it's a huge, let's say, community. And this is close-skin marker recapture. It has been developed in southern Britain tuna, Tunus McCoy, which has specific characteristic because you can sample adults and sample juvenile. There is never overlap. And there is an ongoing effort to develop this method in other species. You have a strong assumption. You really need to sample parents and juvenile, and then it gets really complicated when you have overlapped generation. So the methods cannot be applied to all species, but there is a high potential. And you see the ERC, so the research institution of the European Union, is dedicating a lot of attention, especially to deep sea animal. So you work in deep sea. It has also been applied to elasmobranch, and there are different algorithms that now take into consideration the different aspect of the reproductive. And then this is all about geographical scope. As I say, molecular, yeah, space sounds more like hyperspace, no, like planets. And so these all the applications so far have been on a geographical 2-3 dimension, but we have an additional dimension, which is time. As I said, it's very important to for producing good model that forecast, you first have to indcast. No. Okay. And so we can incast from the past? We have to take historical sample. Again, thanks to the development of technology, we can now analyze DNA from very ancient sample. And then it depends where you put the boundary between historical and ancient. I always forget, and it's also really depends on your study. But you can go back in time, and this is very useful, especially for harvested species, because we have collection. We have scales and otolith collection that has been collected for decades to analyze age and other parameters. And now we have the tool to analyze the really low amount of DNA that can be extracted from an otolith. The otolith doesn't have biological material itself. No, there is no DNA in the otolith. But if the researcher that collected the otolith was not that clean, some biological leftover are on the otolith, and now we have the methods that allow us to analyze the DNA even in such a low and degraded amount, which is the amount of DNA that you can find in old bone or Neanderthal bone, but now we have the genome of Neanderthal or Denisova. A massive sequencing, high-throughput sequencing enable environmental DNA study, but first of all, ancient DNA study. Obviously, first on human. And we can go back in time to find evidence of something that we can see phenotypically, but we don't know if it's also a modification in the evolutionary potential. So if it's just a phenotypic modification, or if it's also a genotypic modification. What I'm talking about, fishery-induced evolution. A colleague, a FishMed previous student, which is, anyone knows Federico Calì? He did a study that demonstrates fishery-induced evolution because he assessed the modification of growth rate and other parameters going back in time. So I think you have seen this case study before. Again, it's code. What happens? The length and age, the size and age of first reproduction now are smaller. Individuals reproduce first, before in time and at smaller size. And what is the implication? Smaller female, less egg, and less reproductive success. So it's really something that phenotypically is observed, and it's important to see if it's just a phenotypical, so it can be reversed, or if it's really something that has eroded and modified their genome, and this cannot be reversed anymore. There are contrasting results. So the idea is that if the survival of large individual is high, so a fishing practice that is not only on large individual, we won't have a fishery-use evolution. But indeed, fishermen, normally we harvest the largest individual, the big reproductor. And so it seems that they start reproducing, so maturation comes early, so that they at least can reproduce a few times. And most of the animals, when they start reproducing, they stop growing. So not all animals have infinite growth, no? And this was possible, we could do it because here, in one of the rooms here, all of a sudden, it's called serendipity, I think. We were informed of wooden boxes coming from, maybe from ex-Yugoslavia, but we are not sure. Come and have a look. And there were these huge boxes. They were huge. They don't seem so huge because the colleague was huge. It was nearly 2 meters. Have you ever met Greg? It was bigger than Adam. And he opened the boxes, and what he found the osteological collection of the Scientific Institute of Rovino. So Professor Massimo Sella, he was a researcher. He was doing studying on migration and connectivity of large pelagic, and he was using a very smart approach. He was using come si dice in inglese? He was using hooks. He knew the different hooks of the different fishing fleet, and when he was finding an animal based on the hook that the animal had inside, he could then reconstruct the path of the animal. Imagine this was the nearly 19th century. So he was really a researcher. He was from a very wealthy family. They own tuna prep. They are the owner of the Banca Sella. Banca Sella is still running. So it's the Sella family, and Massimo Sella was a researcher with a lot of resources, and he had this scientific institute in Istria. Depending on the political situation, he had to flee. He had to escape, and he put everything in boxes and sent it. We don't know, and we don't know how, when, and why they popped up here in Fano. And we were blessed by the opportunity to analyze this. And then we enjoyed really the research field. We really enjoyed talking with the archaeology, fishery historian, storici della pesca, okay? And so we went far and far in the past. The Massimo Sella archive was from pre-industrial area. All the fish were collected around 1910, 1920, but then we needed more back in the time. 700 years were not enough to tell if the genetic erosion was involved. So we started getting along with the archaeologists. And again, it was, have you been there? No. was kind of funny because the only one that could go in this really hot and sunny and fantastic place nearby Tarifa were two Nordic hot suffering colleagues. So they were completely too hot and sweating, and we were all, come on, can you see the beautiful dune and beach in the background? These are the archaeological excavation near Cadiz, at the Gibraltar Strait. So it's Playa de Bologna, Playa de Tarifa, go there. It's beautiful. And so we could extract DNA from archaeological bones. Again, thanks to the ancient DNA lab facility and thanks to the technological improvement. And so we could not only extract DNA, but analyze the bone. And again, a statistical modification of the growth parameter could be inferred. So demonstration of phenotypic fishery-induced or climate-induced evolution. Everything is changing. Now, is climate, is fishery, or what? And we are still digging into the genetic. There is a reduction in genetic diversity. It seems that this reduction in genetic diversity affects more sites in the genome that do have a consequence in the protein that they translate. So really the genome of the species has significant modification over time. Is this due by evolution? Is it due by increased evolution due to climate and anthropogenic impact? It's really difficult to tell apart. And then, okay, so all the blame on the fishery, some of the blame. Aquaculture can help a lot, but we have to do proper aquaculture. So we have been not raising, allevare, rearing animal on land since millennial. We can do, for sure, aquaculture has a potential to provide high quality protein food to the human population, but aquaculture doesn't have to go through all the error of land-based rearing, no? So intensive aquaculture, use of antibiotics or other approaches. And what is also kind of wrong in aquaculture, on land, if your cow or pigs has a disease, you kill them, no? Or if you take care, or if you don't want the disease to be spread. In water, it's more difficult, and sometimes what happens, they release. They say it can happen, incidental release, cages are in the water, you can have storm, cages can get broken, but sometimes you have release. And this is a kind of a problem because then farmed individual that comes from strains that have been selected for certain characteristics that go into the natural environment. And in the natural environment, they reproduce. So it's really important. We can trace with molecular tools, we can trace species, geographic location, a lot of things. We can also trace SKP from aquaculture. Again, this was a project funded by the European Union, and we can really trace and trace back the impact of reproduction between captive and wild animal. And this is again really important for the consequence on the impact. And this is also important for responsible practice, responsible commercial activity. I'm not sponsoring MSc, so Marine Stewardship Council. Who knows what is MSc? Is not the cruise, but is one of the voluntary eco-label for fisheries. You have Friend of the Sea, you have Ecorest, there are different But at the moment, maybe MSC is the most widespread. And we can have eco-label also for aquaculture. There are already products in our supermarket that have this label. Again, it's an eco-label. It's based on the chain of trust. Either you trust it or not. So it's voluntary and depend on you. When you make your choice and you can buy apple 0 kilometer or too good to go. So it's, every time we make choices, no? So if you, I don't think this is the solution. Say radical chic food cannot feed the human population, but when you have the, if you are lucky that you can choose what you buy, maybe we can also think of this, but they have to behave properly, because if they ruin the chain of trust, no.
It's very strict.
Yeah, the 3 pillars.
But I still agree with you about radical shift.
I can tell you my experience. I have one son. My reproductive output is very bad, only one. Not even a very good one, because he was seriously ill when he was born. I was a late mother, so I'm not a fish, so the older I get, not the better offspring. And I was buying everything bio, everything control for my son. And I still try, but then now he likes McDonald's. But I could not afford for the whole family. So I was buying things for him and then for us. So I think a medium family cannot afford to go to Natura Sea, no, for the... So we have, but this is outside genomics. This was a off records. And then detection of pathogens. We are at 10. At 11, I'm done. So we only have 2, and then there is the closing, break is good. But this will be super fast, because who I am to talk about pathogens and microbial, you had already had 2 lectures. One the very first day, and tomorrow. So really, I cannot tell anything more that they will tell you. So again, I was born as a PhD student on microbes. I was scratching microbes out of mosaics in Ravenna. That was my initial PhD. And I was at the Fabit laboratory with Stefano Fedi, the microbiologist, Roberto Borghese. They are? Zannoni. Ok. Cioè, proprio dinosaur, T-Rex. And so you see how different has been my academic career. And then I had some experience on I have some direct experience on identify pathogen in seafood. So identifying pathogens in mussel for commercial, for human consumption, because it's not just a matter of identifying the presence of a pathogen, because microbes, if eumetazoa, if eukaryotes do reproduce and create trouble, imagine prokaryotes, they can exchange genetic material, horizontal gene transfer. So a strain that was not harmful can get the superpower of killing you from another one. So it's not just a matter of checking which microbial strain were in the food, but if those strains were having the gene to encode the toxin that can kill you. Now, do you remember the T-shirt or the meme during COVID? What doesn't kill you mutate and rise again? No? And then when you have all of this, you have the power now, but why things are not perfect? We can tell the species, tell the origin, tell what is in there. So we need enforcement. So with the molecular tool, we can check all the requirements of the ongoing regulation. Maybe the regulation is not the most updated, but for sure, when you go to when you sell some fishery in the broader sense product, you have to tell from where, with which fishing gear, okay, the species, and if it's fresh or processed and so on. With molecular, you can tell all of this, not the fishing gear, sorry, but at least which species and where. But now you need enforcement, either enforcement or you can apply this tool to support certification. If a Friend of the Sea is a certification body based in Italy, they're all Italian, if Friend of the Sea certified a fishery, it's good that they can provide some proof of evidence. Look, my fishery is really doing this. I can show you proof with DNA. I can really tell you if this fish has been caught in English Channel, North Sea, or somewhere else. And again, now with the genomics, you can tailor small assay. You don't need to go and make the genome of every fish also, because it will take 6 months to have the answer and the fish is. So you need something very fast, very easy. So you can go back from genomics and cherry pick the part of the genome that really have the power to tell you what you need. And you can go as low as less than 10 assay. They cost less than 10 euros, and you can really trace individual to species and geographical origin. Now we have commercial chip. So back to my chip, which are not the chip I was doing my PhD, but you have commercial assay that you can buy for all the zootechnic species. So pig, pork, cow, but also sea bream, sea bass, meager. So for aquaculture species, you have this chip that can provide information genome-wide. But for example, Scandinavian countries have received the high funding from ICES and other institutes to develop a chip not for aquaculture species, but for fishery species. So now they have a chip that can genotype herring, sprat, and a lot of species to provide information on all these aspects. Obviously, running the analysis at the very end is not not expensive, but producing the background information to reach the resolution to develop such a chip requires a lot of effort. So why we are not doing things perfectly? Because there is still a problem in uptaking all this information, no? The role of genetic under the EU Common Fishery Policy. Can we really estimate how management can really benefit? So you see a lot of, we have the potential to give the answer. Are these answer uptake by the end user? So really very recent paper, no? Gene, fish and fishery, translating science into policy. So there is really, now we need the next step. And this is a nice paper. So again, people working best place for the best science, but also Jan Martinezson is at the JRC, so is the European Union. Why we don't apply genetics to improve? Is still too costly? Ok, stop. Sono stata veloce, no? We take a short break, then we discuss the essay, and then I'm ready to take all the question on the credits. 10 minutes, we start again at 1. No, dobbiamo finire all'1:30. E c'è modo di bloccare la registrazione?
Sempre. No, quelli in basso.
Da qua? Ah, scusami, esatto.
Interrompi la registrazione.
Ah, interrompo, ok.
Esatto.
E dopo
Adesso.
facciamo
E poi
il commento.